For as long as humans have, well, farmed and raised animals, we've been subtly but pretty powerfully shaping nature. Think about it. Every dog breed, every kind of apple, the livestock we eat, they're all products of generations and generations of selective breeding. We picked organisms with traits we liked, bred them, and slowly, over centuries, kind of guided their evolution. It's an incredible success story in itself, really. It gave us the food that feeds the planet. But yeah, that process is undeniably slow, really slow. What if we could fast forward that, make it, incredibly precise and rapid?
That's exactly the question scientists started asking back in the 1970s and 80s. That was when recombinant DNA technologies really started taking off. It was a huge leap for science. The core idea was pretty revolutionary. Identify a specific gene for a trait you want, make copies, clone it, and then introduce that gene directly into a new organism. The potential for agriculture seemed, well, enormous.
Yeah.
Imagine, higher crop yields, using far fewer pesticides, maybe even foods with better flavor, more nutrition.
And that promise, it pretty quickly started turning into reality, didn't it? That first genetically modified food approved for sale, the Flavr Savr tomato, that hit the market back in 1994. It was engineered to stay firm and ripe longer. And after that, we saw other things pop up, like papaya and zucchini resistant to viruses, canola oil with different fatty acids, and then the really big ones, like corn and cotton engineered to resist insects, or soybeans and sugar beets that could tolerate common herbicides.
Huge impact crops.
But almost right away, this new tech sparked some really intense debate. On one side, you had people seeing this vast potential, maybe a way to tackle malnutrition globally or even help crops survive climate change. On the other side, though, there was some vehement opposition. Sometimes people even took, well, drastic measures to stop these new varieties being introduced. So as you can probably guess, when it comes to genetically modified foods or GM foods, there's a lot of, let's say, hyperbole and, frankly, misinformation flying around from all sides.
Right. And that's precisely why our mission with this deep dive is to try and cut through some of that noise. We're going to introduce you to the basic science behind GM foods, look at the promises they hold, but also the problems and challenges they present. And we'll definitely explore the controversies surrounding them. Our goal isn't to convince you one way or the other. It's really just to present the facts based on our source material so you can evaluate these pretty complex questions for yourself and hopefully walk away feeling genuinely well-informed.
Okay, so let's start right at the beginning. What exactly are genetically modified foods? It sounds straightforward, but the definition, it can actually be a bit slippery.
That's a really good place to start. Yeah. So when we talk about genetically modified organisms, GMOs, we generally mean organisms whose genomes, their DNA, have been altered in ways that don't happen naturally through mating or recombination. Now, selective breeding, like you mentioned, is technically a form of genetic modification over long periods. But the common definition of GMOs, the one people usually mean, refers specifically to organisms modified through genetic engineering, often using those recombinant DNA technologies we talked about.
Okay, so genetic engineering is the key difference here from traditional breeding.
Exactly. It's incredibly precise. It lets scientists clone and transfer one or more specific genes from one organism to another. And this can be done within the same species or even between totally unrelated species. It also lets us alter an organism's own genes, maybe to make them more active or less active. So if a gene gets transferred between unrelated species, say a fish gene into a plant, that resulting organism is often called transgenic. If the gene transfer happens within the same species, like moving a gene from one type of potato to another, the term cisgenic is sometimes used.
Transgenic, cisgenic. Got it.
And it's also really important to remember that biotechnology is a much, much broader term. It covers all sorts of things, like using enzymes and detergents or the processes for making wine or cheese. Genetic modification is just one tool, one application within that huge field.
And these GM crops, they're not just a small experiment anymore, are they? They're really a global phenomenon. They're grown in about 30 countries, covering something like 11% of the world's arable land, which is quite a bit. But here's where it gets really interesting. Almost 90% of all that GM acreage is concentrated in just 5 countries: the US, Brazil, Argentina, Canada, and India.
The big agricultural players.
Yeah, and the US alone accounts for roughly half of that global acreage. Think about this. Here in the US, a staggering 93% of soybeans and 90% of the corn or maize grown are from GM varieties. And that filters down to us, the consumers. It's estimated that over 70% of processed foods in the U.S. contain ingredients derived from these GM crops.
Wow, 70%, that's huge.
It really is. Now, while there are around 200 different GM crop varieties approved here in the U.S., only about 2 dozen are actually widely planted. They mostly focus on traits like herbicide tolerance, insect resistance, things like that, though some are for nutrition too.
And it's not just plants we're talking about. In 2015, the AquAdvantage salmon became the first, and actually still the only, GM food animal approved for us to eat.
Ah, yes, the fast-growing salmon.
That's the one. It's an Atlantic salmon, but it was engineered to grow twice as fast as normal Atlantic salmon. It reaches market size in about a year and a half versus the usual 3 years. The modification involves adding a growth hormone gene from a Chinook salmon, but it's controlled by a genetic on switch, a promoter, from an ocean pout, which is the type of eel. This switch keeps the growth hormone gene active year-round, not just in warmer months.
So it just keeps growing.
Pretty much.
Yeah.
Now, the company behind it, AquaBounty, has put safeguards in place. They've committed to only selling female eggs that are also triploid, meaning they have 3 sets of chromosomes instead of 2, which makes them sterile. And the fish are grown in contained, inland facilities, land-based tanks, with filters and barriers to prevent escapes into the wild.
Okay, so they're trying to be careful.
Definitely. But critics still raise concerns. They worry that the sterilization method might not be absolutely 100% effective all the time. And they worry about potential kind of downstream effects if any of these GM salmon did manage to escape and interact with wild salmon populations.
Right, that potential environmental impact. It really raises an important question, doesn't it? How do we balance this incredible potential for innovation with, the necessary precautions to protect the environment?
It's the central tension in a lot of these discussions.
So with all these different GM foods emerging, what were the main driving forces behind their development initially? If we look at the 2 most common types of GM crops, the answer seems pretty clear. First up, we have herbicide-resistant GM crops. These make up about 70% of all GM crops grown globally today. That's a huge percentage.
It really is.
And the most common modification here involves introducing a gene, usually from a bacterium, that makes the crop plant tolerant to a broad-spectrum herbicide like glyphosate. You probably know it as the active ingredient in Roundup. Now, for farmers, this is, it was a game changer. They could spray their fields with glyphosate to kill weeds while the crop itself was growing unharmed. That's often more efficient and cheaper than, say, tilling the soil repeatedly or using multiple different herbicides. It can also reduce soil damage from less tilling, and some argue it has a lower environmental impact compared to using several older, potentially more toxic herbicides.
Right. Those were the key selling points.
Then, the second most common type of GM modification is insect resistance. And this brings us to the really fascinating world of Bt crops. They're named after a soil bacterium, right? Bacillus thuringiensis.
That's exactly right. Bacillus thuringiensis, or just BTP for short, is this amazing soil bacterium that naturally produces proteins called cryproteins, crystal proteins that are toxic, but only to specific groups of insects. Things like caterpillars, certain flies, beetles, some bees, and wasps.
So how does it actually work?
Well, for the toxin to have any effect, the insect has to actually eat plant material containing these Bt proteins. Once inside the insect's gut, which has a high pH, the protein crystals dissolve. Then, enzymes in the gut cut the protein into its active toxic form. This active toxin then binds to very specific receptor proteins on the surface of the insect's gut cells.
Okay.
This binding disrupts the gut cell membranes, basically poking holes in them. The gut breaks down, the insect stops feeding, and eventually it dies.
Wow, it sounds pretty targeted.
It is incredibly targeted. That's what's remarkable. There are over 200 different types of these cryproteins known, and each insect species has unique gut receptors that will only bind to a few specific Bt proteins. So scientists can pick a beet strain that produces a protein highly specific to a major pest, say the European corn borer, while leaving beneficial insects like ladybugs or bees largely unaffected.
That's clever.
And it's worth noting, broad itself isn't new in farming. Spores from the broad bacterium have been used as a natural insecticide for decades, often sprayed on plants in both conventional and organic gardening. They break down quickly in sunlight and soil, and studies show they don't harm groundwater, mammals, fish, or birds.
So how do they get it into the plants?
That's the GM part. Scientists identified and cloned the specific cry genes that code for these insecticidal proteins. Then, using genetic engineering techniques, they introduce these genes directly into the plant cells. So the resulting broad crop plants actually manufacture their own burdocry proteins in their tissues. When the target pest insect takes a bite, it gets a lethal dose.
Ingenious, really. Self-protecting plants. Now, while most of these big GM crops, the herbicide-tolerant, insect-resistant ones, are mainly used for animal feed or processed ingredients like corn syrup or soy oil, there are some GM foods developed specifically for direct human consumption. And perhaps the most famous, and you could argue the most controversial example, is golden rice.
Ugh. Golden rice. Yes, it's definitely well known and highly debated. It aims to tackle a really critical global health problem: vitamin A deficiency. This is a serious public health issue in over 60 countries, particularly across parts of Asia and Africa where rice is the main staple food. The World Health Organization estimates that globally, around 190 million children under 5 and 19 million pregnant women are deficient in vitamin A.
And the consequences are just devastating, aren't they?
They truly are. Somewhere between 250,000 and 500,000 vitamin A deficient children become blind every single year. And tragically, about half of those children will die within a year of losing their sight. Vitamin A is also absolutely crucial for a healthy immune system. So deficiency makes people, especially children, much more vulnerable to severe infections and other diseases.
That's just awful.
It is. And current efforts to fight it, like distributing high-dose vitamin A supplements or promoting home gardening for leafy greens, they've had some success, but their reach is often limited by cost, logistics, and consistent access.
So golden rice was proposed as another potential tool, right? How does it actually work?
Well, the interesting thing is that rice plants naturally make beta-carotene, which our bodies convert into vitamin A, in their leaves, but they don't make it in the part we eat, the grain, the endosperm. The grain actually has the precursor molecule, the starting material, but it's missing a couple of key enzymes needed to finish the biochemical pathway and produce beta-carotene.
So the machinery is partly there, just incomplete?
Exactly. So for golden rice, scientists essentially gave the rice plant the missing genetic instructions. They inserted 2 genes. One came from a common soil bacterium called Erwinia iridivora, and the other originally came from daffodils in the first version, golden rice 1. These genes provide the instructions for the missing enzymes. They also carefully attached genetic on-switches, promoters, to these genes to make sure they were only turned on in the endosperm, the edible part of the grain.
And that made the rice yellow.
Yes. The beta-carotene gives the grains a distinct yellow-orange color, hence the name golden rice. Now, that first version, golden rice 1, it worked, but the beta-carotene levels were relatively low, maybe providing 15–20% of a child's daily vitamin A needs.
Okay, a start, but maybe not enough.
Right. So then came golden rice 2. This was a significant improvement. Scientists swapped out the daffodil gene for a similar gene from maize, from corn. This simple switch resulted in a massive, more than 20-fold increase in beta-carotene production in the grain.
Wow, a 20-fold increase. That's huge.
It was a major breakthrough. And what's really fascinating and crucial is that human clinical trials have shown that the beta-carotene golden rice 2 is efficiently converted into vitamin A by the body. It's estimated that just about 150 grams of uncooked golden rice 2, that's less than a cup, could potentially supply all of a child's daily requirement for vitamin A.
That's incredible potential. So where is it now?
It's currently undergoing extensive field testing, biosafety assessments, and efficacy trials required for regulatory approval in several countries. The plan, if approved, is to make the seeds available to resource-poor farmers at the same price as conventional rice varieties. And importantly, farmers would be allowed to save seeds from their harvest to replant the following year, which addresses some concerns about farmer dependency.
That seems like a key point.
It is. But despite this clear potential humanitarian benefit, controversies around golden rice definitely persist. Critics raise concerns about, say, promoting reliance on a single staple food or potential unforeseen long-term health or environmental effects. These are complex issues we should definitely unpack a bit more.
Okay, so now that we know what some of these GM foods are and why they were developed, let's maybe pull back the curtain a bit and look at the how-to. How do scientists actually get these new genes into the plant cells?
Right, the mechanics of it. There are 2 main methods used for creating GM plants, and both usually involve starting with plant cells grown in a lab in what's called plant tissue culture. You can take small pieces of plant tissue, grow them on a nutrient medium, and they form a mass of undifferentiated cells called a callus. These calluses can then be stimulated to grow roots and shoots, eventually regenerating into a whole new plant.
Okay, so you start with cells in a dish, then how do you get the new DNA in?
One way is a physical method called the ballistic method. It's often just called the gene gun.
A gene gun? Sounds dramatic.
It kind of is. You basically coat tiny particles of heavy metal like gold or tungsten with copies of the DNA fragment containing the gene you want to introduce. Then you use this device, the gene gun, to fire these DNA-coated microprojectiles at high speed into the plant cells or tissues. It's literally blasting the DNA in. Some cells survive this bombardment, and in a fraction of those, the introduced DNA might get integrated into one of the plant's chromosomes.
Huh. Seems a bit brute force.
It is, but it can work, especially for plant species that are harder to transform with other methods. But the arguably more elegant and widely used method leverages a natural expert, a soil bacterium called Agrobacterium tumefaciens.
Agrobacterium. I think I've heard of that. It causes tumors in plants.
Exactly. It causes a disease called crown gall disease, where the plant forms tumors. Agrobacterium does this because it's a natural genetic engineer itself. It carries a special piece of circular DNA called a tumor-inducing plasmid, or T plasmid. When the bacterium infects a plant wound, it transfers a specific segment of this T plasmid, called the tDNA, directly into the plant cell's nucleus, where it integrates into the plant's own chromosomes.
So the bacteria naturally injects its DNA into the plant.
Precisely. The genes within this tDNA then hijack the plant cell's machinery to produce nutrients for the bacterium and cause the tumor growth. Scientists figured out how to harness this natural delivery system. They essentially disarm the T plasmid by removing the tumor-causing genes from the tDNA segment. Then, they insert the desired gene, the one they want to introduce into the plant, into that same tDNA region.
Clever. So they use the bacteria's own tool against it, sort of?
Kinda, yeah. They use its natural ability to transfer DNA. They then let these engineered agrobacterium infect the plant cells in tissue culture. The bacterium does its thing, transferring the modified tDNA containing the gene of interest into the plant genome. Of course, for the introduced gene to actually work properly in the plant, it needs to be cloned next to the right control sequences, like a promoter, which acts as an on switch, telling the cell when and where to express the gene, like only in the endosperm for golden rice, and a terminator sequence, the off switch.
Right. It's not just the gene, but how it's controlled. Now, you mentioned that even with these clever methods, getting the DNA successfully integrated and working, that transformation rate, it can be pretty low, right? Like maybe only one cell in 1,000 actually gets transformed properly?
That's often the case, yes. The efficiency can vary a lot depending on the plant species and the method, but it's rarely high.
So if you've got this dish with maybe millions of plant cells and only a tiny fraction have actually taken up the new gene, how do you find those few successful ones? Growing them all into plants would be impossible.
Exactly. You need a way to select only the transformed cells, and that's where selectable markers are absolutely crucial. They act like a sieve or a filter.
Selectable marker. Okay, how do they work?
There are two main types. One is called negative selection. Here, along with your gene of interest, you also co-introduce a marker gene that provides resistance to something that would normally kill plant cells, like an antibiotic, say hygromycin, or an herbicide. You then grow all the treated cells on a medium containing that selective agent, the antibiotic or herbicide. Only the cells that successfully receive both the gene of interest and the resistance marker gene will survive and grow. The untransformed cells die off.
Okay, so you kill off the ones that didn't work.
Right, that's negative selection. The other approach is positive selection. This often uses a marker gene for an enzyme that allows the plant cells to utilize a sugar source that they normally cannot metabolize, like mannose. So you put all the cells on a growth medium where mannose is the only available sugar, the only carbon source. Untransformed cells can't use mannose, so they starve. But the cells that successfully took up the gene of interest and the special enzyme marker gene called phosphomannose-isomerase, or PMI, can now thrive on the mannose medium.
Ah, so instead of killing the failures, you specifically feed the successes.
Precisely. And what's fascinating here is that this positive selection method, using the PM gene, was actually the selectable marker system used to create golden rice too.
Oh, interesting.
Yeah, and subsequent studies on the PMI protein produced by this marker gene have shown it's easily digested, it's non-allergenic, and non-toxic in animal feeding tests, addressing some potential concerns about marker genes remaining in the final food product.
Right, that makes sense. So maybe to really nail this down, let's quickly walk through golden rice 2 again, but focusing on how these methods actually came together.
Sure, good idea. So for golden rice 2, the scientists needed to get 3 genes into the rice genome. They needed the crittii gene from the bacterium Erwinia uritivora, the improved Cy gene from maize, corn, and that selectable marker gene, PMI, which came from E. coli.
Okay, the 3 key components.
Right. They carefully cloned these 3 genes, each with its own specific promoter, on switch, and terminator, off switch, into a single Ti plasmid vector that modified Agrobacterium shuttle plasmid we talked about. called PSYN12424 in this case. For example, the Psy and Critii genes, the ones for making beta-carotene, were put under the control of a rice gluten promoter. This promoter is specifically active only in the developing endosperm, the part of the rice grain we eat. That ensures beta-carotene is made there, and not, say, in the leaves or roots.
Very precise targeting.
Exactly. The PIA marker gene, on the other hand, was linked to a maize polyubiquitin promoter, which is active in pretty much all plant tissues, ensuring selection could work effectively.
Got it. So they assembled this custom DNA package.
Yes. Then they used agrobacterium containing this engineered PSYN12424 plasmid to infect rice cells taken from embryos grown in tissue culture. After the infection step, they transferred all the cells to that selective growth medium containing mannose as the only sugar source. Only the rice cells that had successfully incorporated the tDNA carrying all three genes, CRUTI and PLI, could survive and grow on this medium.
The positive selection step.
Correct. These surviving cells were then stimulated with plant hormones to form calyces and then coaxed to regenerate into whole rice plants. Finally, they analyzed these regenerated plants using molecular techniques like PCR to confirm that all three genes were indeed present and properly integrated into the rice genome. And of course, they selected the plants whose grains show the highest levels of beta-carotene production, visible by that intense golden yellow color.
It really illustrates how these different tools, tissue culture, agrobacterium, specific promoters, selectable markers, all work together. It's quite a sophisticated process.
It truly is. It showcases the ingenuity involved in genetic engineering, combining knowledge of plant biology, microbiology, and molecular genetics.
And that ingenuity, it hasn't stopped, has it? The field keeps moving, evolving at, well, breakneck speed, it feels like sometimes. And here's where things get really interesting now. We're starting to talk about the next wave, often called genome editing. Things like ZFNs, TALENs, and especially CRISPR-Cas systems. These methods seem poised to change the game entirely.
They absolutely are changing the game. The key difference, the really fundamental difference with genome editing techniques like CRISPR, is their incredible precision. They allow scientists to make very specific, targeted changes directly to an organism's existing DNA sequence. This could be changing just a single DNA letter, a nucleotide, or deleting a small specific gene sequence, or inserting a new sequence at a precise location.
Okay, so it's like using a word processor to edit the DNA, rather than just inserting whole new chapters.
That's a pretty good analogy, yeah. And crucially, many of these edits can be made without introducing any foreign DNA from another species into the organism's genome. You're essentially just tweaking the organism's own genes.
Ah, and that has big implications for rules and regulations, right?
Huge implications. Because in many cases, no foreign genetic material is involved. Many regulatory agencies, including the USDA of the United States for plants, have so far decided that organisms modified using some of these techniques don't fall under the same regulatory framework as traditional GMOs that contain transgenes.
So they might get to market much faster.
Potentially, yes. It could dramatically speed up the development and commercialization process, and it's definitely shifting how we think about, define, and regulate genetic modification in agriculture.
And we're already seeing real-world examples, aren't we? Like that calyx potato, what did they do there?
Right, the calyx hyalix soybean was actually their first product, but they also developed a potato. Using TALENs, another genome editing tool, they inactivated a specific gene, one coding for an enzyme called vacuolar invertase. This enzyme normally breaks down sugars when potatoes are stored in the cold. These sugars can then lead to undesirable browning and the formation of acrylamide, a potential carcinogen, when the potatoes are fried.
So by knocking out that gene?
By inactivating that gene, the potatoes accumulate less of these sugars during cold storage. The result is a potato with a longer shelf life that also produces significantly less acrylamide when fried. Better for storage, potentially safer for consumption.
That sounds genuinely useful. And there was a mushroom, too?
Yes, the CRISPR-edited mushroom. This was back in 2016. Scientists at Penn State used CRISPR-Cas9 to knock out or inactivate a gene for an enzyme called polyphenol oxidase, or PPO. PPO is the main enzyme responsible for that browning reaction you see when mushrooms, or apples, or potatoes for that matter, are bruised or cut.
Right, the annoying browning.
Exactly. By inactivating one of the PPO genes, the edited mushrooms resist browning much longer, giving them a longer shelf life and making them look more appealing to consumers.
And the USDA didn't regulate that one.
Correct. The USDA determined it didn't require regulation under their rules at the time, precisely because the editing process didn't introduce any foreign DNA. It just made small deletions in an existing mushroom gene. It was seen as similar to mutations that could potentially arise naturally or through conventional breeding, just achieved much faster and more precisely.
Fascinating. And this isn't just for plants, right? Genome editing is being used in farm animals, too.
Absolutely. The potential applications in livestock are also enormous. For instance, researchers at the Roslin Institute in Scotland, famous for Dolly the sheep, are using genome editing, specifically CRISPR, to develop pigs that are immune to African swine fever virus. They're doing this by introducing very small, precise changes into a pig immune system gene called RDLA, aiming to mimic the natural resistance found in wild relatives like warhogs.
Wow, tackling a major animal disease like that.
Exactly. And elsewhere, for example, at Seoul National University in South Korea, scientists have used genome editing to create double muscle pigs. They did this by inactivating a gene called myostatin, MSTN. Myostatin normally acts as a brake on muscle growth. By disabling this brake, the edited pigs develop significantly more muscle mass, leading to higher lean meat yields. Similar edits have been made in cattle, too.
So potentially more meat with less feed.
That's the idea.
Yeah.
Now, while these genome-edited animals are currently often falling outside the traditional GM regulatory frameworks, just like the plants, this is a very dynamic area. It's quite likely that new regulations specifically addressing genome editing in animals will be introduced in the near future as the technology becomes more widespread.
It really feels like we're entering a new era of genetic technology. But despite all these scientific advances, these new tools, GM foods and maybe genome-edited foods, soon to remain perhaps the most contentious products of modern biotechnology. The debate is just incredibly polarized, isn't it? You have advocates talking up the benefits, increased farm productivity, less pesticide use, conserving soil, feeding growing populations, and then you have critics raising serious concerns, claiming these foods are unsafe for humans, harmful to the environment, or both.
It's definitely a polarized landscape.
Yeah.
And this brings us back to what is perhaps the single most crucial point for you, the listener, to take away today. The term GM food itself is a massive generalization. It's simply not scientifically valid or helpful to make sweeping statements about the safety or impact of all GM foods.
Right, because each one is different.
Yeah.
Exactly. Each genetically modified organism, whether it's blight corn, golden rice, or that AquAdvantage salmon, is a unique product. It has specific genes altered or inserted using specific methods, resulting in specific traits. Comparing them all under one umbrella is like comparing, I don't know, a bicycle and a jet plane, just because they both have wheels and are forms of transport. Blanket statements about GMO safety or GMO impact just don't hold water scientifically.
That's a really important clarification. Each needs to be looked at individually.
Precisely. Each construction is unique and therefore needs to be assessed on its own merits and potential risks. This nuance is absolutely critical for any informed discussion.
So when we try to deconstruct these controversies, keeping that nuance in mind, two main areas constantly surface. First, human health and safety. Advocates often point out that GM foods currently on the market have been consumed by billions of people and livestock for over 20 years now, with no credible, documented evidence of adverse health effects linked specifically to their consumption. They also highlight that the vast majority of animal feeding studies required by regulators, often hundreds of them, have shown no negative health effects. And they tend to argue that the few studies claiming to find negative effects have often been criticized by the broader scientific community for having flawed methodologies or interpretations.
Right, the Seralini paper on rats and Roundup Ready corn was often cited as an example there, which was later retracted by the journal, then republished elsewhere, but remains highly controversial regarding its design and conclusions.
And advocates also sometimes point out that human clinical trials aren't typically required for foods developed through other methods like conventional breeding or mutation breeding, even though those methods can also introduce significant and often unknown genetic changes and potentially novel proteins or allergens into our food.
That's a fair point about regulatory comparisons.
But critics push back strongly on the health issue, don't they?
They do. They often counter that relatively few GM foods are actually eaten directly by humans in large quantities as whole foods. Much of the GM crop acreage goes into animal feed or is processed into ingredients like oils, starches, or sugars, where the novel DNA or proteins might be largely removed or denatured during processing.
So maybe direct human exposure isn't as high as the acreage suggests.
That's one argument. Critics also emphasize that there have been very few long-term human clinical trials specifically designed to assess the health impacts of consuming GM foods. Golden Mice 2 being a notable exception with its efficacy trials.
Right.
And they argue that many of the required animal feeding studies are relatively short-term, often just 90 days, which they suggest might not be long enough to detect potential chronic or subtle long-term health effects if they were to exist. The debate really centers on what constitutes sufficient evidence for safety.
Okay, so that's the health debate. The second major area of controversy revolves around environmental effects. Critics often cite both documented and potential negative consequences for the environment, which they argue could also indirectly impact human health down the line. Advocates, on the other hand, argue that GM crops, particularly beet crops, have actually led to an overall reduction in the use of broad-spectrum chemical insecticides. And they maintain that potential environmental risks associated with GM crops can be identified, assessed, and managed through appropriate stewardship practices.
It's another area with strong points on both sides.
And here's where it gets really fascinating, because we're seeing real-world evolution happening right before our eyes, driven by how these crops are used. We're talking about the emergence of resistance, both herbicide resistance in weeds and insecticide resistance in pests.
Exactly. This is a major challenge.
Take herbicide resistance. Since glyphosate-tolerant crops like Roundup-ready corn and soy were widely adopted in the mid-1990s, we've seen a dramatic increase in glyphosate-resistant weeds. I think the number is over 24 different weed species identified as resistant in the U.S. alone now.
That's right. Palmer amaranth, water hemp, horseweed. Some of these have become huge problems for farmers, seriously impacting crop yields and requiring more complex and costly weed management strategies.
And why does this happen?
Well, it's basic evolutionary pressure. When farmers rely heavily, sometimes almost exclusively, on a single herbicide like glyphosate year after year, it creates intense selection pressure. Any individual weeds that happen to have a natural mutation conferring some level of resistance survive the spraying, reproduce, and pass that resistance gene on. Over time, resistant populations dominate the field.
So it's evolution in action, accelerated by the technology.
Precisely. Though, it's crucial to point out, as you hinted earlier, that herbicide resistance is not unique to GM crops. Weeds have evolved resistance to virtually every class of herbicide ever developed long before GMOs came along. Over-reliance on any single weed control tactic will eventually lead to resistance.
Good point. And we see a similar story with insect resistance.
Yes, unfortunately. Since bright crops were first introduced in 1996, there are now documented cases of field-evolved resistance to blight toxins in at least 8 major insect pest species globally, including significant pests like the corn rootworm and the cotton borer.
So the bright crops are becoming less effective against some key targets.
In some regions, for some pests, yes. The selection pressure is intense. If the insects are constantly exposed to the same bait toxin, any rare individuals with mutations allowing them to survive will be strongly selected for.
So what are farmers and scientists doing to combat this resistance problem? They can't just keep planting the same bite crops indefinitely, right?
No, that's clearly not sustainable. Several strategies, often called insect resistance management or IRM plans, are being employed. One major approach is developing and deploying GM crops that produce 2 or more different beet toxins simultaneously. This is sometimes called pyramiding or stacking toxins.
The idea being it's much harder for an insect to evolve resistance to 2 different toxins at the same time.
Exactly. The odds are much lower. Another absolutely critical strategy, especially for beet crops, is the implementation of refuges.
Refuges, what are those?
A refuge is basically an area of non-GM crops planted near or within the beet crop field. This refuge area must be large enough to allow a substantial population of susceptible insects that are not resistant to the beet toxin, to survive and reproduce.
Okay, so you're deliberately keeping some non-GM plants around for the bugs. Why?
It sounds counterintuitive, but it's key to delaying resistance. The idea is that any rare resistant insects that emerge from the beet crop field are likely to mate with the much more numerous susceptible insects emerging from the refuge. If the resistance trait is recessive, which it often is initially, then the offspring of these matings, heterozygotes, will still be susceptible to the beet toxin in the GM crop. This dramatically slows down the spread and buildup of resistance genes in the overall insect population.
Ah, so the refuge dilutes out the resistance genes.
That's the principle. Maintaining effective refuges is mandated by regulatory agencies like the EPA in the U.S. as a condition for planting beet crops. However, farmer compliance with refuge requirements has sometimes been inconsistent, which can accelerate resistance development. Farmers are also encouraged to use integrated pest management (IPM) strategies, combining beet crops with other control methods like crop rotation, monitoring pest levels, and using conventional insecticides only when absolutely necessary.
It sounds like managing resistance requires a really integrated and careful approach. Okay, besides resistance, what's the other major environmental concern we hear about? Gene flow.
Yes, gene flow is another significant area of discussion and research. This refers to the potential transfer of the engineered genes, the transgenes, from the GM crop into populations of non-GM crops, or even into sexually compatible wild relatives growing nearby.
How would that happen? Pollen drift.
Primarily through pollen drift, yes. Wind or insects can carry pollen from a GM crop field to fertilize plants in adjacent non-GM fields or wild populations, if they're close enough, and flower at the same time. Seeds accidentally spilled during transport or planting can also lead to GM plants growing outside cultivated fields.
And there are documented cases of this happening.
Oh yes, there are numerous studies documenting the presence of GM plants, like herbicide-tolerant canola or sugar beets, growing in uncultivated areas like roadsides, field margins, or even around ports and processing facilities, sometimes quite far from where they were originally planted.
So what's the worry if these genes do spread?
The main concern is the potential for outcrossing the successful hybridization between the GM crop and a compatible non-GM crop or wild relative. This could lead to the unintended presence of the transgene and the trait it confers in populations where it's not desired.
Like herbicide tolerance spreading to a weedy relative, making it harder to control.
Exactly. That's a major concern. Or, for instance, if you're an organic farmer or a farmer growing non-GM crops for specific markets that require them to be GM-free, gene flow from a neighboring GM field could contaminate your harvest, causing economic losses.
I remember reading about that escaped GM bent grass in Oregon.
That's a classic example. Creeping bentgrass engineered for glyphosate resistance escaped from experimental field trials in Oregon several years ago. Because it's wind-pollinated and perennial, it established itself in irrigation ditches and other areas outside the test plots and proved very difficult to eradicate, precisely because glyphosate was no longer effective against it.
And the environmental impact could be bigger if the transgene actually gives the wild relative some kind of advantage, right?
Yes, the concern is amplified if the transgene provides a trait that increases the fitness of the wild relative in its natural environment. For example, resistance to insects or diseases, or tolerance to drought or salt stress. This could potentially allow the wild relative populations carrying the transgene to become more invasive or disrupt natural ecosystem balances. However, whether this happens in practice depends greatly on the specific gene, the specific wild relative, and the specific environment.
So what's being done to try and contain this gene flow?
Regulators and researchers are exploring various containment strategies. One common approach is requiring mandatory isolation distances, buffer zones, between GM fields and compatible non-GM fields or wild populations to reduce the likelihood of cross-pollination. The required distance varies depending on the crop's pollination mechanism, wind versus insect, and biology. Other more high-tech biological containment methods are also being researched. One idea is using RNA interference, RNAi technology, which can specifically silence genes to engineer GM plants that are male sterile, so they don't produce viable pollen.
No pollen, no gene flow that way.
Right. Another approach is called chloroplast transformation. Instead of inserting the transgene into the plant's main nuclear genome, you insert it into the DNA found in the chloroplasts, the plant cell's energy factories. In most major crop plants, chloroplasts are inherited only through the egg cell, not through pollen. So genes located in the chloroplast DNA wouldn't be spread via pollen drift.
Interesting. Are these methods widely used yet?
These biological containment methods, like sterility or chloroplast transformation, are still largely in the research and development stage. They present their own technical challenges and haven't been widely implemented in commercial GM crops yet, but they represent potential future strategies for managing gene flow concerns.
Okay, so we've covered a lot of ground here. From the ancient practice of selective breeding, through the development of the first GMOs, the science behind how they're made, the rise of genome editing, and the really complex controversies surrounding their use. So what does this all mean for you listening right now? We've really tried to explore the immense promise, the intricate science, and yeah, the complex realities and debates surrounding genetically modified foods.
And hopefully, what's become clear is that understanding the underlying science, and especially embracing that crucial nuance, that each GM organism really is unique and needs individual assessment, is absolutely vital for evaluating this rapidly evolving field. As we always say, knowledge is most valuable when it's understood and applied critically. And that critical thinking is just essential in today's world, which is often overloaded with information and sometimes misinformation.
So maybe a final thought to leave you with, something to mull over. Given the incredible, almost unbelievable precision of these new gene editing tools like CRISPR, tools that allow us to make tiny, targeted changes to DNA, and considering the ongoing, very real global challenges of food security, climate change impacting agriculture, and a growing population, how do you think our relationship with agricultural genetics, with deliberately shaping the crops and animals we rely on, how might that evolve over the next, say, 10 or 20 years?
Yeah, it's a huge question. Will the potential benefits, disease resistance, enhanced nutrition, climate resilience, ultimately be seen to outweigh the perceived risks?
Or will public perception, the social and ethical debates continue to be the most significant factor, maybe
Yeah.
even the biggest hurdle, for the adoption of these powerful technologies? Something to think about. Thank you so much for joining us on this deep dive into the fascinating and definitely complex world of genetically modified foods.
We really hope you feel more informed and maybe more empowered to engage with this important topic in your own way.